Okay, this is part 2 of the lecture on meiosis, and we're going to talk about the sources of can't spell meiosis. Meiosis. The sources of variation in meiosis that allow you basically to look like your parents but not identical to your parents. So you might have characteristics like you might have your mom's nose but your dad's eyes. And there are 3 sources of genetic variation. Sources of genetic variation. One is called independent orientation or independent assortment. I prefer assortment. And this is at metaphase 1. Okay. The second is the random fertilization of gametes. And we'll go over these individually, of course. And 3, the crossing over or the exchange of genetic material during prophase, during synapsis. During synapsis. Okay. I'm going fairly quickly because I know you guys can pause the video and rewind it, so I'm just going to kind of just go. And then of course, if you have questions, the Zoom meetings would be an appropriate time to ask those. If you need me to re-explain something, I'll save these. We can watch the video together if we need to. And I can go into more detail. Okay, so number 1, independent assortment at metaphase 1. So remember at metaphase, the where's my picture? This is from last time. At metaphase 1, we're talking about this, the homologous chromosomes are lined up along the midline, right? So I'm going to kind of change that a little bit. And to simplify this, possibility B, we're just going to look at 2 homologous chromosomes, or 2 sets of homologous chromosomes. So let's make the red chromosomes are the ones you get from your mom, and the blue ones are the chromosomes that you get from your dad. Okay? So we'll make one kind of big one and one kind of small one. And then the homologous one that you get from your mom. So one way that these could line up during metaphase is you'll have your dad's on one side of the cell and the smaller one on the other side of the cell, and then your mom's on one side and then the smaller one on the other side, okay? The other way these could line up is they could be on the same side. like this. So moms are all on one side and dads are all on the other side. So what's going to happen when this cell divides into 2? So remember, during meiosis 1, the first cell division, the homologous chromosomes separate, okay? Not the chromatids. So in this cell here, you're going to have the big copy of the chromosome from your mom, oops, and the small chromosome from your dad. And in this cell, you're going to have the big chromosome from your dad and the small chromosome from your mom. Okay, so they're mixed, right? Here, you're going to have, both those cells are going to have either moms or dads. And then when they divide again to make gametes, so this is during meiosis 1. And then this is meiosis 2. Those are going to divide. So these are the gametes down here. You can probably see where I'm going with this. This is going to have one copy from the dad, the big one, the small one from your dad, the small one from your mom, small one from your mom, and this is going to be the big one for your mom, big one for your mom. So there's this recombining or mixing that can happen, or big mom, little mom, big mom, little mom, big one for your dad, little one for your dad. Okay, so this gamete is going to have all of dad's chromosomes. This gamete is going to have all of mom's chromosomes. This gamete is going to have one from mom and one from dad. So you get this mixing up of the chromosomes. Okay, so that's the independent assortment of the chromosomes that happens at metaphase 1. Of course, this is just with 2 chromosomes, and we have 23, right? So with this combination, you have 1, 2, 3, 4 different possibilities of blue, red, red, blue, red, red, blue, blue, right? So that's 4 different combinations that can combine with just these 2 chromosomes. We have 23. So this is a little beyond the scope of the class, but to just kind of explain this, the probability of those combinations, you can think of this as like flipping a coin. So here are 2 quarters. What's the probability of getting heads if I flip this 1 quarter? So getting heads once is 50% chance, right, of getting heads. On 1 coin. What's the probability of me getting heads on this quarter? It's also a 50% chance. What's the probability of getting heads on both? So both quarters. It's a little tricky. If you've taken statistics, then you would know the answer to this, but a lot of people haven't really thought about this. In order to get the probability of getting heads on both, you multiply the probabilities together. So the probability of getting heads on this quarter is 50%. Probability of getting heads on this quarter is 50%. The probability of getting both, you multiply those together. So the chances of getting heads on both is 25% on both. Okay. So we can use this probability, getting back to this, what's the probability of getting red and red? it's the probability of getting red on here and red on there. And you can do the math. The way this works is you do the number of possibilities. So on this quarter, or on this, the probability of red and red, you've only got two options here, right? So you do 2 times the number of coin flips. We've got 23 chromosomes. So if we do 2 to the 23rd power, again, this is just a little bit beyond what we're going to be covering on the exam, but just to kind of show you how this works. The probability of getting all your dad's chromosomes in one sperm, let's say, or all your mom's chromosome in one egg is 2 to the 23rd power, which ends up being 8,388,608, a 1 in 8 million and change chance of getting all one parent's, say dad's, chromosomes in one gamete. So there are 8 million and change possibilities, different variations of sperm, 8,388,000 different variations of possibilities of eggs. So the odds of that happening are really, really rare. Of you looking identical to your dad is like a 1 in 8.5 million chance of that happening. So with that, we can go to source number 2, the random virtualization of gametes. So the principle behind this is eggs and sperm, they randomly fertilize each other. Each other. Okay. So you have 8,388,608 sperm variations, 8,388,608 egg variations. The probability of getting an identical mom and dad fertilization is 8,388,000 times 300. So it's like basically almost impossible. I mean, it is like almost impossible. So that's the take-home message here, is that the eggs and sperm randomly fertilize each other, and it increases the variability of each offspring from the parents. So let's kind of take a side step here real quick and talk about variations of genes. And for an example, we're going to look at mouse characters. And let's look at the coat or the fur color and the eye color. And we're going to name these genes just kind of arbitrarily: gene C and gene E. Okay. Big C, we're going to say, if you have this version of the gene, if you have big C, it has brown fur. If it has little c, it has white fur. Big E is going to have black eyes, and little e is going to have pink eyes. Okay. So let's draw this out. So say from mom, and we'll draw these. Red is again mom. We'll have a chromosome and let's say it has, here's the gene for fur color, and mom has big C and big E. Dad we're going to say dad has the gene for white fur. Well, it's here too. So these are identical copies of the chromosome, right? So dad has got white eyes, little c, and, I'm sorry, pink eyes and white fur. And mom's got brown fur and black eyes. Okay, so let's, if we separate these during meiosis, all the gametes, and there's no recombining at all the gametes are either going to be little c, little e, or they're going to be big C and big E, right? All the gametes are going to be one of those. But that's not what happens in real life. Remember, the chromosomes will cross over and exchange segments of them, right? So the third source of variation is in crossing over. During synapsis. And this is the swapping, let's say, or the exchange segments of DNA or chromosome to, say, mix up the genes between mom and dad. Mom's and dad's chromosomes in your gametes. Okay. So let's go back to our mice. So this is the chromosome that a mouse has inherited from its mom. And it has big C and a big E rather. And when the homologous chromosome from dad lines up during synapsis, little c, little e, there's going to be a breakage. And let's say it breaks there, and those two pieces are going to swap. So they break. And then they form what's called a chiasm. Let's go up like this. So that structure right there is called a chiasm or a chiasma. That's the location. So the chiasma is the location of crossing over. Then they will reform with the exchange of that genetic material. So now mom's chromosome, or the one you got from your mom, has got a little segment of the section that you got from your dad attached to it. And then same thing, dad's now has a little section of the chromosome that your mom gave you. Okay? So that's crossing over. And now, if we look at this again, we've got big C, big E, and now we've got dad's little e on that chromosome. And then down here, that's a big C. This is little c, big E, little e. So if we go back to what we did when we were talking about this, and we're going to start making gametes with these different combinations. So let's draw that again. We'll draw, let's draw like this. So the gametes that are made, one variation is going to have, you can't see both of those. There we go. So let's just look at the gamete that's made with this particular chromosome. It's going to have big C, big E. So these are the gametes. One possibility is it's going to have that combination of those genes. Another possibility is it's going to be mixed. It's going to have, excuse me, big C, little e. Another combination or possibility is going to be little c, big E. And then the last possibility is going to be little c, little e. Okay, so this is again another source of variation where you might have your mom's nose, but your dad's eyes. Here, you might have your mom. You would have your mom's eyes and your mom's. I'm sorry. What was I saying? The eyes and nose. So this might be your mom's nose and your mom's eyes, your mom's nose and your dad's eyes, your dad's nose and your mom's eyes, or your dad's nose and your dad's eyes. So you can have these combinations that can, basically present themselves in the offspring. These two that you have both moms and both dads, these are called parental types. This is a parental. These two are called recombinant. Recombinant types. Okay. So again, 3 sources of variation. The independent assortment of the chromosomes at metaphase. So depending on which way they're flipped during metaphase. The random fertilization of the gametes. And then finally, crossing over during synapsis. Okay, I'm going to stop there and pick up part 3 and maybe even part 4 coming up. Okay.